This is lecture 18 in Principles of Metabolism course. In this lecture we'll look a bit at the compartmentalization of metabolism and some transport phenomena. So compartmentalization, that's a tricky word. So what is this? This has to do with metabolic compartments. So this can happen on many levels, but basically it's ways of closing off a space where you can have reactions happen. And of course, cells themselves are metabolic compartments. And it's one of the theories of why cells exist. It's because it's a way of closing off a space where you can do reactions more efficiently. And of course, inside cells we have organelles, and organelles also represent compartments that tend to isolate reactions and metabolites and specialize in doing particular types of reactions. So what's shown here is a summary figure of a genome scale model of human metabolism that was published in 2007. And the numbers that you see within each organelle is the number of reactions, which tells you that at least in this kind of reconstruction and modeling of metabolism, we have two very large compartments, and cytosol is the largest by far. Mitochondria also contains a large amount of metabolic reactions. And there's a fair number of reactions which are specialized in other compartments. So what could be the benefits of this? Why would you like to isolate reactions in several different compartments? Well, one thing could be that it might be difficult for a couple of reactions to sort of coexist together in a single compartment. They might use the same substrate or act in opposite ways and would sort of be fighting against each other if they were in the same compartment. But you might want to carry them out in two separate places. And then there's also the property that these organelles can sort of have different environments. So some of them can favor different types of reactions. So we tend to say that the cytosol, for example, is reductive. So meaning that this is an environment where we have usually high concentrations of NADPH, so an electron carrier that tends to drive reductive reactions. So a lot of reductive biosynthesis happens in this compartment. It's good for that. We tend to say that mitochondria are oxidative, meaning that we have a high concentration of NAD in its oxidized form, meaning that that can drive an oxidative reaction. NAD can become reduced, and so it can oxidize the substrate. So mitochondria tend to be sort of set up to do oxidative reactions, which of course fits well with their role as the sort of energy powerhouse. And then there are some other sort of compartments with specialized chemistry. So the peroxisomes do particular oxygen chemistry. We have seen that there are some fatty acid oxidation reactions that require this type of oxygen peroxide type of chemistry. So that's sort of isolated to that compartment. Lysosomes are acidic, so we can sort of sequester the special reactions that require low pH in that compartment. The endoplasmic reticulum, the ER, is of course an organelle that contains a lot of membranes, so a membranous organelle. So therefore there's a lot of lipid biochemistry that goes on in the ER. So a lot of lipid synthesis reactions happens in those membranes. And so on. So this is another dimension of metabolism, you could say, the compartmentalization, and it is certainly a very important part of how metabolism is organized. So here's a particular example of compartmentalization. This is a very busy slide with lots of details, but we'll just look at a couple of aspects. So this is one carbon metabolism. This is the metabolism using folate as a carrying factor, or tetrahydrofolate in the active form. And what it does is that it carries this type of one carbon unit. So this is a methylene, for example. This is a formyl, but still one carbon with a sort of aldehyde type of group. And these one carbon units are used for purine synthesis or for methylation and stuff like that. But there's a kind of curious system here where this particular metabolism is compartmentalized into cytoplasm and mitochondria. And it is thought that this is because of the oxidative reductive properties of mitochondria and cytoplasm. So in order to be able to drive this system of reactions in the oxidative direction, to be able to take these one carbon groups and oxidize them down, there's a set of enzymes that exist in the mitochondria and which are driven by NADH. And it's thought that that will drive these reactions in the oxidative direction. And then in the cytosol, we have the same sort of complement of reactions. So these ones and these ones are the same, but these ones have specialized and using the NADPH cofactor in this case. And it is thought that this will drive these reactions in the reductive direction in the cytosol. And so therefore these pathways can be sort of adapted to do different things in the two compartments. You could also think of other types of compartments, which are not organelles, but smaller kind of environments or structures. So we often talk about microenvironments where you could have very specialized things happening locally. So localized metabolism. And there are several examples of this in the structure of the cell. There are particular pockets or niches where you could have particular metabolic events happening. And this can be efficient because you will have a much smaller space and you can sort of cluster some reactions together and they can perform a little pathway, and their substrates and products are not going to diffuse away, so you can accomplish a complex metabolic task efficiently in one place. And you can direct maybe energy to different processes and things like that. So what's shown here is an example, a recent example of glycolysis, so particularly two steps of glycolysis, the GAPDH and the phosphoglycerate kinase steps. So this is the energy yielding step of lower glycolysis. So these enzymes have been found to co-localize together with these vesicles that move around in very rapid transport in the axons of neurons. And so they help move vesicles around. And so this is an example where you have this particular part of the glycolytic pathway localized here so that you can produce ATP, which is locally used by these kinesins move around vesicles. So in that way, you can direct a metabolic product to a particular location in the cell. And this you also can think of as a kind of compartmentalization, because this ATP is not going to go anywhere. It will be immediately consumed, and therefore, in effect, this is closed off. This ATP is local and cannot be used by other processes. There is also this concept of substrate or metabolite channeling. So in a way, I guess the example with the vesicle is a case of this. But it's often thought of as a way for pathways to assemble locally. Sometimes these are enzyme complexes, and it's a bit of a gray area if a metabolite is being channeled between two different enzymes, or if we should just consider it an intermediate in a more complex enzyme. So, whichever way you want to put it. But there are these examples where you have enzymes sitting together and are carrying out the pathway locally. And again, this helps because substrates won't diffuse away, and so you will be able to do a complete pathway more efficiently. There's a lot of examples of this in bacteria, maybe not as much in human or eukaryotic cells, but probably just has to do with the fact that it's harder to study in some ways. One example that does exist is the step from malate to oxaloacetate to citrate in the TCA cycle. So these are two enzymes that are thought to exist together. You see very low concentrations of oxaloacetate, which is free. So we think that it's being channeled directly from the dehydrogenase to the synthase. And you can do various types of kinetic studies and stuff like that to come up with evidence that there is channeling going on. It's not very easy to do, so there is always some debate about whether this exists or not. But I think the consensus is that there are this type of systems. This compartmentalization phenomenon also causes some problems for us when we try to study metabolism. And so often we are not able to separate compartments, subcellular compartments, when we want to measure metabolites. So we tend to get overall measurements or mixtures of what's in different compartments. And this is just something that is worth keeping in mind, that it can be hard sometimes to tease out the actual contributions of different organelles to the overall measurements that we have. And sometimes you can have high concentrations in one particular organelle, but you can have a smaller amount of that particular, the volume of that organelle. And these are things, of course, combine together. And so you can have high local concentrations which are not visible in the final measurements because they are high concentration but in small volumes. And so these type of ambiguities we have when we measure metabolites in general from cells. This also appears when we do isotope tracing. And in this case, what will happen is that isotopomers could exist in different states, in different organelles. You might, for example, produce this metabolite called M in a different way in an organelle, say the mitochondrion, than you would do in the cytosol. They might have two different sources. So you might have a labeled isotopomer appearing in the mitochondrion, an unlabeled one in the cytosol. But of course, if you just mash this up and do a cell extract and measure this on a mass spec or something like that, you will see the mixture. And this can be misleading in some cases. There are ways to deal with this in terms of modeling. You can model your measurement as a mixture of two unknowns. And if you have enough data points and not too many unknowns, then you might be able to tease this apart. There are some studies where people have tried to do this experimentally by, say, rupturing only the plasma membrane and get those metabolites out and spin away the mitochondria or something like that. But it's quite hard to do experimentally. So this is a challenge for studying metabolism, that we have compartments. Now, between all of these compartments, there must be some kind of transport events. And this is an interesting way of controlling metabolism, to control the traffic between these compartments. So in addition to the control we might have on the level of enzymes, this is another mode, you might say. And probably one that hasn't been studied as much, so we don't know so much about these systems. But we know that there are different kinds of transporters and they act in different ways. So first of all, in some cases we just have non-selective type of pores, just openings in membranes. The nucleus is one example where you have the nuclear pores, which means that the nuclear environment is probably the same as the cytosol, at least with respect to small metabolites that easily go through these pores. So these those two compartments are communicating pretty closely. In other cases, you have very sort of closely walled off compartments, like the mitochondrial inner membrane is not permeable to many things. So there you need to have selective transporters that let things go in and out. And then depending on what kind of these transporters that a cell chooses to put in a membrane, you can of course control what can go through. And well, one classic example is the glucose transporter at the plasma membrane, the insulin-sensitive glucose 4. Choose to put that there when you want to take up glucose, but there are many other examples. But again, this is a less studied mode of how metabolism is regulated and functioning, but probably an important one. So you can have transport events that are completely passive. That in itself can regulate things because you allow things to pass or not. But then we have also the active transport cases, and these can occur in many different sort of flavors, and sometimes we don't, it's not so easy to recognize that it's actually active transport, but it might be. The most obvious ones are the ATP-driven pumps type of transporters, and ABC cassettes and stuff like that. And so these are just simply ATP-driven, and so you can build up an ion gradient that way. So those are easy to recognize. But there are also lots of transporters that are coupled to counter ions in various ways, and so it means that if you have an existing gradient of a counter ion, then you can utilize that to transport something else. So you could have a situation like this, where you have a gradient of sodium across the plasma membrane, for example, with the sodium potassium ATPase, and then you use sodium, its gradient, to bring something else in or out of the cell, some metabolite X. So you could recognize that, okay, we know that sodium is an important gradient across the plasma membrane, so if you see a transporter that is co-transporting with sodium, then you can figure out what it does. But it can get more complicated. There are transporters that are coupled to the gradient of another metabolite, which in turn is coupled to the gradient of sodium, which in turn is coupled to ATP. When you have enough of these systems in a membrane at the same time, it starts to get difficult to figure out what is actually happening. So you see a lot of transporters that are coupled to major metabolites like glutamate and aspartate and things like that. And so it can be a bit tricky to figure out what are the gradients between the organelles and how are these things actually driven. So this is an interesting but challenging part of metabolism. Then there is also the phosphorylation way of trapping things inside membranes. We talked about this in relation to glycolysis, where we quickly phosphorylate sugars as soon as they come into the cell. And that's a way of trapping them in the cell, we said. And this is actually a way of building up a gradient. So in order to sort of capture all the glucose, in this case, that you would take up into a cell, you use the phosphorylation strategy. And so here we are relying on a kinase, which again ultimately is using ATP, to make sure that we have a kind of gradient. We have much more of this product than we have of the substrate, because we're using ATP to make sure that the reaction is driven far in this direction. And so that's one way of creating a kind of gradient, or should we call it maybe an imbalance between the substrate and products. But again, it's a way of doing that so that we can keep this metabolite low, so that we in turn can just pull in more metabolites from the outside, just by the Le Chatelier kind of law. And so again, this is actually a kind of way of active transport, and it costs ATP in the same way that an ATP-driven pump would cost ATP. So these are all ways of transporting metabolites against the gradient. And cells need to do this in many situations to concentrate nutrients on the inside so that metabolic reactions can occur efficiently. So I think the field of transporters in metabolism is still it's still early days. We don't know so much about this. A lot of proteins have certainly been studied in detail, but the big picture is not clear, I think. So what we know from the biochemical characterization of these transporters is that it's frequently the case that a metabolite can have many transporters that accepts that metabolite. And conversely, any given transporter can accept several other metabolites. So you can look at this in databases like the human-seq database. And so if you go and look at any metabolite of interest, in this case I picked serine, it turns out serine is transported by a bunch of these different transporters. And if you look at each of those will be connected to other type of transport events. So all of these on the left side. And so you tend to have this type of more unspecific kind of networks between metabolites and transporters. So compared to enzymes, it generally seems that transporters cannot have the same type of specificity. And this makes it difficult to analyze this part of metabolism in a way. So I think this is a field that is on the rise, but I think it is still early days for transport phenomena.